Hi, and thanks for joining us. My name is Colin Merrifield, and I'm a product manager with Bio-Techne. And today we'll be talking about challenges in scaling analytics for cell and gene therapies. So for those of you not familiar with Bio-Techne, we're an umbrella brand encompassing a number of specialist life science companies where we've been growing extremely quickly in the last several years. We've been involved in a number of mergers and acquisitions, and this has allowed us to build a broad and comprehensive portfolio of technologies, reagents, and services. And really within the last several years, we've pivoted our attention to developing better solutions to support the cell and gene therapy space. And we've asked our brightest and best scientists and engineers to think about how we can help this part of the life sciences industry scale and eventually provide a better quality of life for the many patients which it's bound to support. One area of pharmaceutical development from which we derive a lot of inspiration has been the evolution of therapies in the modern times. So in that time, we've seen therapies morph from the relatively simple small molecule to the classes of therapeutic antibodies and therapeutic proteins, and then more recently to what we're familiar with as therapeutic viruses, nanoparticles, and cell therapies. And one of the things that we take away from this is that we need to make sure that analytics evolve alongside and in parallel to the therapies. And what's occurred to us is that a lot of the predicate methods that are used for analysis of these therapies have not changed much within the last several decades. And we want to be at the forefront of this and ensure that we're always helping to reduce the sample requirements needed for analytical techniques. We're reducing the operator time and inputs that's needed, and that the result is a much higher quality data set on the back end. And alongside of the platforms, we also want to ensure that these analytics are supported by an ecosystem of scalable reagents and materials. So we're proud to offer a comprehensive portfolio of cytokines and growth factors, small molecules, media, and supplements. And we really believe that the stability of this ecosystem also contributes to the overall data quality of the analytical tools and platforms. A specific part of cell and gene therapy research and development that we'll focus on today are analytics for viral vector production in three areas in specific: vector titer, vector purity, and vector characterization. And with that, I'll give a brief technology introduction for three of the platforms that you'll hear about from our speakers today in a bit more depth. The first is our Simplex technology line and our Ella platform, which has taken the traditional immunoassay or ELISA and reformulated it into a microfluidic cartridge. So within this cartridge, we have all of the reagents, and the entire assay is controlled by the microfluidics. And some of the benefits of this is a large reduction in the amount of hands-on time that's needed to execute the assay. So only about 15 minutes of preparatory work. We've also been able to reduce the sample volume that's needed that's involved with running the immunoassay to just 25 microliters. Another platform that you'll hear about today is our Maurice platform, which is part of our ICE technology line. And our ICE technology line is a gold standard in the characterization of monoclonal antibody therapeutics and is excitingly finding new opportunities for characterization of proteins within the viral vector production process. So with Maurice, we're able to inject a protein within a capillary, separate it by its charge, and then quantitate the different variants that we observe with really high fidelity in terms of the resolution of the technique. This takes as little as 50 microliters, and setting up a run takes as little as 45 minutes of hands-on time from an operator. And the third platform, which you'll hear about today, is our Simple Western technology line. And our Simple Western technology is a reimagination of your traditional Western blot within a microfluidic capillary. You have a matrix within that capillary which provides for separation, either based on size or charge. The sample is then immobilized and then can be probed with different antibodies. And this provides, similar to the simple Plex technology line, a drastic reduction in the amount of sample volume that's needed to execute a Western blot, just 3 microliters. And it also greatly simplifies the workflow with just 30 minutes of hands-on time, followed by a completely automated execution of the assay. And some of the feedback that we've gotten from our customers within the space in terms of the benefits that they've realized from our platforms has been, the impact of the reduced sample volume has been highly enabling. They can use microliters instead of milliliters to execute these techniques. The high level of automation and the drastic reduction in operator time has been really important for their teams to be able to focus on higher value activities and have a greatly improved bandwidth. And the cartridge consumables and the enclosed nature of the platforms is also particularly important when we're considering analytics for the viral vector production process, as this helps to control viral contamination within a lab. And sort of that enclosed nature of the platforms in conjunction with a high level of automation also contributes to really high quality data, so we can control for a lot of the variables that traditionally complicate analytical methods, and this generally helps to increase the data quality that the systems produce. And downstream of that, all 3 of these technology lines are supported by a comprehensive set of CFR compliance tools that allow you to that support your data integrity needs within whichever regulatory landscape you inhabit. And we really feel that all of these benefits in conjunction are helping our customers iterate their research and development faster, and ultimately getting a product to market faster. And with that, I'll thank you again for joining us, and it's my distinct pleasure to introduce our first speaker, Dr. Kiren Baines. And Kiren is a bioprocess scientist with eXmoor Pharma Concepts. Welcome, Kiren.
Thanks, Colin, and it's an absolute pleasure to be invited today and present today. So I'm going to give you an introduction to eXmoor Pharma, who we are, and then a brief introduction to AAV and their process development workflow. And then I'll spend the majority of the time showcasing the analytical challenges when doing process development, and how the Ella platform has really been revolutionary to the challenges we face day to day with bioprocess sample and bioprocess analytics. So at eXmoor Pharma, we work in the cell and gene therapy field. So we design GMP facilities for cell and gene therapies and biopharma, but we also help with their manufacturing bioprocess development challenges. So our team, which at the moment consists of 20 plus scientists, are able to take really early stage clinical manufacturing processes and optimize the entire process, so from upstream to downstream, but with tailored analytics. And the goal is really to produce an improved, efficient, and robust process. So currently, we're based in the southwest of England, and we're actually undergoing a really exciting phase of the company. So we have purchased and we'll be moving to a new facility, which you can see there in the top right corner of the slide. And we also plan to have GMP capabilities from 2023. So we currently have a global footprint and have worked with over 130 companies, over 20 countries, and we look forward to expanding on this in the future. So now I'd just like to give you a brief introduction to AAV. So as you're all probably aware, adeno-associated viruses are a promising therapeutic modality for monogenetic diseases. So these viruses are capable of delivering genetic sequences of up to 5,000 base pairs to non-dividing cells, which is unlike lentivirus. They're non-integrating. So in addition to this, they are also shown to offer durable protection. So this is another reason adding to the popularity of AAV. So here you can see a number of different AAV serotypes and the target tissues that these serotypes target. So at eXmoor Pharma, we have experience with working with a number of these different AAV serotypes, and I've just highlighted a few that we have extensive experience working with. So we've got experience working with AAV2, 3, 5, 8, and 9, and we look forward to expanding on this in the future. So it probably goes without me saying that AAV is one of the largest classes of gene therapies. So, when you start out, quite often early stage gene therapy companies use techniques that are quite common to the academic research lab setting. And this is what you can see on this slide. So, you can see techniques here, such as adherent cell cultures, which make use of FBS. And you can also see along the workflow here that there's freeze store lysis cycles and also ultracentrifugation. So this starting point, or the as is process, really isn't optimized for a therapeutic products. So the aim when developing a process is to improve the manufacturing scale, robustness, and cost. However, this all needs to be achieved whilst maintaining, or actually achieving, a necessary level of quality and safety. And as you can imagine, this quality and safety is fundamental for your therapeutic product. So, quite often, the as is process does not meet these requirements for clinical or commercial products. So, we look to develop a more suitable to be process which can meet these objectives. And so, to meet the quality, safety, and cost considerations, this often involves a workflow, which you can now see on the bottom half of the slide. So, this workflow often involves developing suspension processes which use chemically defined medium and also chemical lysis. Following clarification, you also tend to use capture and polish chromatography steps, and then finally diafiltration to formulate your product. So when planning process development, there are a number of activities which need to take place to successfully realize these goals of quality, safety, and cost. So here I'm showing an example workflow of the types of studies required from early stage development to late stage development. And you'll notice at the top, the banner workflow is the same to what I presented in the earlier slides, and really does follow along the lines of early stage development to late stage development. So the type of studies you may look to work more into, for example, may include for expansion, cell line evaluation. For production, you may look to optimize transfection or culture conditions. For harvest, you might look at screening different lysis conditions and buffers. And then for purification, this may include optimizing chromatography steps such as capture and polish. And then finally, for your formulation and fill, you may look to carry out formulation screening studies. Now, in order to select the most suitable or optimized conditions, a significant amount of analytical measurements are required. And here on the graph to the bottom left, you can see the number of upstream samples in dark green and the number of downstream samples in light green, following the workflow I presented on the previous slides. And in fact, when we looked at what this number actually equates to, it's above 800 samples. All of these samples require analysis on a number of different analytical platforms. So I think you'll agree with me, there's no small task. So here I'm showing different types of analytical platforms that are important when it comes to characterizing your bioprocess workflow, but also your clinical or commercial product. So it's important to appreciate that it's not possible to develop a process with only one analytical method. You need a number of orthogonal analytical methods to understand key aspects of your process. So, for example, it's important to understand and determine your capsid number. And typically, this is done using ELISA. But it's also important to understand the proportion of capsids that are full. And typically, this is done using qPCR or ddPCR, which is more accurate when it comes to quantifying viral genome concentrations, and i.e., the capsids that are full. So over the next few slides, I'm going to look at the importance of determining capsid titer and how the ELA platform has really been instrumental to our process development. So as I mentioned, ELISAs are commonly used for physical titer quantification. And in bioprocessing, they are widely used to understand product concentration to optimize the process, but also to look at recovery across each unit operation. ELISAs have also been traditionally used because they're quite good at dealing with impure or low concentration samples, which other analytical platforms, such as gel-based techniques, would struggle with. So although there are a number of advantages to the ELISA, there are also a number of challenges when it comes to carrying out an ELISA. So one of which I've demonstrated here with the schematic, and this encompasses extensive hands-on time and limited throughput. So we all know it takes roughly 5 to 7 hours to complete a traditional ELISA. So that's an entire day to get data back. And you may require data quite quickly as you're making in-process related decisions. An ELISA also has quite a narrow dynamic range. And typically this means you have to run a number of dilutions for your sample, again, limited in the amount of throughput. And here I'm just showing a schematic of an ELISA plate map representation. And you can see a number of dilutions are required. And actually, if we use the example of 800 samples, this would mean you need to run 80 to 100 plates to achieve analysis of those 800 samples. So overall, there are a number of limitations that slow down in-process related decisions. Now, the ELA platform overcomes many of these limitations faced by the ELISA, and these points are highlighted here on this slide. So they include greater dynamic range, high throughput, and greater precision. And over the next few slides, I will demonstrate the benefits of the ELA platform over the ELISA using real-life process development examples. So one of the challenges in process development is that there is a broad range of capsid concentrations across each unit operation. So you can see in the schematics to the left that the range consists of 10 to the 10 to 10 to the 15 across the entire workflow. And often we're changing parameters to improve titer and recovery throughout the whole process. So what this means is that usually there's low confidence in the concentration of a given sample. Now, because the ELA has such a higher dynamic range when compared to the ELISA, and you can see this here with the graph in the middle. So you can see the ELA is shown in blue and the ELISA in orange. The broad dynamic range means that concentration uncertainty of your sample becomes less of a problem as you're targeting a much broader range now compared to the ELISA. So here I'm showing two different sample types, so crude samples and purified samples, and 3 different samples were taken of each of these sample types. And this was all run on one ELA cartridge and actually using only one dilution across all the samples. And on this cartridge, we were able to capture both crude samples, which have a much lower capsid per mil concentration compared to purified samples. This means that we can go in with fewer dilutions and be really confident that even with these fewer dilutions, we're going to target the concentration of our sample. So overall, running fewer dilutions and allowing us to make decisions across a broader range of process samples is really adding value to our process related decisions. So, as I mentioned earlier, a challenge in process development is the vast number of analytical samples. And so, throughput is a critical parameter to making informed process decisions. So, we often run AMBER 250 miniature bioreactors, and you actually can see a picture taken from within our lab of the AMBER 250 bioreactors on the left there. And we often purify these bioreactors to assess changes upstream has on downstream. So for example, we might want to assess changes different culture media has on the process workflow. So we typically compare the titer of 6 bioreactors and assess recovery across the process workflow. So I'm showing this as a schematic there on the right of the slide. So with this sort of experiment, we generally typically generate around 30 samples, and you can see where these samples are generated across the workflow on the slide. So to analyze these samples on the ELISA, we would have to run a multiple plates. And in fact, you can see there that we'd have to run 3 ELISA plates. So what this does is introduce plate variability, which we know from experiments within our lab that this variability ranges from 10 to 20%. Now, if we were running these samples on the ELA cartridge, because it has such a higher throughput compared to the ELISA, we would actually only require 1 ELA cartridge. And the schematic there is of a typical plate map we would generate for the ELA cartridge. So running all the samples on this 1 cartridge means we completely eliminate any influence of inter-assay variability. So this again is allowing us to make more informed decisions of our process development workflow and samples, and also allowing us to create a smart design of the workflow. So the final case study I'd like to go through is looking at the whole process. So for example, here in the slide, I'm using 50-liter bioreactor and looking at upstream all the way to downstream, and looking specifically at this slide of running multiple batches. So there's several reasons why in process development you may require to run batches. And the example I'll use is, for example, you want to look at the difference between 4 different constructs, and you want to look at this difference across the entire workflow. So if you were to do this, you would typically generate 13 samples per batch and times that by 4 for each batch. So this means you'll have a large number of analytical samples. And actually, when you figure out how many ELISA plates that requires, it's 4 ELISA plates compared to 1 ELA cartridge. So therefore, as mentioned previously in the previous slides, again, we're removing inter-assay variability. So not just is it increasing our precision, but also our efficiency to make process-related decisions. So in conclusion, process development leads to a generation of a large number of samples. One of the key challenges is throughput and precision. And the ELA platform has really been instrumental to increasing our overall throughput, and by doing so, allowing us to make more efficient and accurate process-related decisions. So thank you for your attention.
Thanks so much, Kiren, for a really, really excellent talk. And with that, I'll introduce our second speaker, Dr. Chris Heger, who is a Director of Application Science here at Bio-Techne. Welcome, Chris.
Hi, thanks, Colin, for the introduction, and thanks, Kiren, for the a great talk showing how Ella can be used in characterization of AAVs. I'm going to take it a step further and talk about how some of our other technologies can be used to leverage a better workflow for analysis of viral vectors. So in addition to Ella, Bio-Techne has a number of capillary electrophoresis instruments that can support the entire development pathway. So this is kind of a more simplified view of what Kiren was showing, but if we start essentially with cells at the beginning and purified AAVs at the end, our Simple Western and Maurice technologies play well on either the early stage shown here for Simple Western or the later stages of product characterization and QC with Maurice. To summarize how Simple Western is applicable to the AAV purification workflow, I'm summarizing a collaboration here between ourselves, the Cell and Gene Therapy Catapult, and ProGen. The Cell and Gene Therapy Catapult leveraged Simple Western technology for real-time analysis of their AAV2 purification process. The summary of this work was put into an application note, which is shown here on the right. I'm just going to show a little bit of that data today. So using Simple Western to monitor the AAV purification process at the Cell and Gene Therapy Catapult started with the use of our total protein kit. The total protein kit on Simple Western, you can think of like a Kumasi staining. It will label all of the proteins that are separated and captured in the capillary. As we move from the left-hand side of this image to the middle, we're essentially going from the flow-through or the lysate all the way to the elution fractions that are in the middle here. So this is a great way to get individual identity of the viral proteins in your AAV, but it's also important to be able to understand that where those viral protein signals are really coming from, and we can do that more specifically leveraging the immunoassay capabilities of Simple Western. So using a VP123 specific antibody from ProGen, the Cell and Gene Therapy Catapult reran the same samples. And you can see a much cleaner picture here because we're only detecting the very specific VP proteins. So taken together, Simple Western is able to provide you an idea of purity as well as the identity of your AAV samples throughout the purification. Taking it a step further, if we look at the elution fraction from this process and compare the total protein assay on the top and the immunoassay on the bottom, you can see that using both assays together is actually quite valuable. The total protein assay detects a single impurity slightly lower than 40 kilodaltons that is missed in the immunoassay detection, and that's presumably because this impurity is not VP protein related. In the application note, we took this a step further by using VP-specific antibodies to leverage on the platform for more specific detection of individual VP proteins. In the top panel is our VP123, in the middle is a VP12 specific antibody, and on the bottom is a VP1 specific. So in addition, we're able to provide information on the individual viral proteins. Last month, we released a brand new application note where we're taking the AAV analysis further with our JESS platform. In this application note, we're leveraging the ultra-sensitive total protein detection at our JESS instrument, as well as our replex feature. So the total protein assay, as I've described, labels all of the proteins in the sample. So we're going to use that again, but in this particular case that I'm going to describe, we're going to turn up the labeling concentration to enhance our detection capability. In addition, we're using replex in this application note, which is essentially like stripping and reprobing the capillary, but it's completely automated within the process. So using this replex feature, we're able to look at a series of AAV process samples and have an idea of their sense of purity. So shown here on the left is an overlay of the two detections. So the first stage of detection is with an anti-VP1, 2, 3, and then the second, or the replex detection, is of the total protein in the sample. And that's shaded in gray, the blue trace behind the samples. You can see that for AAV1 and AAV4, we have essentially no detection in the immunoassay, but significant total protein in those samples. And conversely, AAV7 and AAV9 are both well detected with this antibody, as expected by its reported immunoreactivity. The exciting thing about this assay using the 5x total protein approach is we're only using 50,000 copies of an AAV per well, which is extremely attractive given how challenging it can be to get enough sample for your in-process analytics. Using the Compass software, we can easily also quantify the specific viral protein peak areas, as well as the total protein in these samples. And that total protein information can certainly be used automatically in our software to normalize the loading between these samples. Lastly, in the application note, we went head-to-head with Cypher-Ruby staining. So Cypher-Ruby staining for SDS-PAGE is typically used to look at impurities or other low concentration samples, and can typically detect around 1 nanogram of protein at its lower end of detection. We took an AAV2 sample, 1 × 10⁻13, and serial diluted it from 1 to 5 to 1 to 40, and then ran it on an SDS-PAGE, followed by Cypher-Ruby, and imaging on one of our Fluorochem imagers, or running on our JESS platform. Shown here is the Cypher-Ruby, where we can detect easily in the 1 to 5 all 3 VP proteins, but as you get towards the 1 to 40, you can only essentially see the VP3 peak. So what we found through this work was that the Simple Western assay is actually more sensitive than Cypher-Ruby when you use the 5x total protein labeling. So using equivalent loading, 3 microliters per well, here's the data for the Simple Western. And zooming in a little bit closer on that 1 to 40 dilution where the Cypher-Ruby seems to fall off, and that's that top image in the middle. The Simple Western assay still has a lot of room in the detection capability at 1 to 40. And that's further realized when we tried to quantify, and we could measure AAV2 signal with as little as 160-fold diluted AAV. So I think it's really a key of this application, though, is that we're able to detect lower levels than Cypher-Ruby, so we should be able to completely automate any needs of that level of detection at this point. So as Colin mentioned, in addition to Simple Western, we also have Maurice, which is also a CE instrument, and has both IEF and CE-SDS detections. And Simple Western is a CE-SDS instrument as well, with immunodetection, and Maurice is a direct detection instrument. So Maurice is widely accepted for use in the biopharmaceutical arena for purity analysis for CE-SDS, and as I'll also talk about, for charge heterogeneity. It's a fast platform with only about 35 minutes per sample, and you can handle up about 48 samples per batch, and it's quite reproducible. We've been spending about the last year working on AAVs, and we've published a few application notes. The first one highlighted here, and this is summarizing our work with AAVs on CE-SDS. Just diving briefly into a little bit of that data. When we started this project, we bought some AAVs commercially, and this was the SDS-PAGE that came with them. So you can see the 3 VP proteins, but they're fairly hard to appreciate, and there are, of course, some other peaks that are even harder to see. So we ran this sample on our Maurice CE-SDS platform, and here's the lane view from that result. So it's much crisper, much easier to detect all of the proteins as well as the impurities below. Now this lane view is backed up by a data-rich electropherogram shown here. In black is that AAV sample, and in blue is a blank. So we're able to accurately measure the VP proteins and then everything else in the sample and get a sense of the purity, which in this case was only about 70%. But to show you that we can look at better samples, I guess I could say, I'll move on to some preclinical AAVs in a moment. The method is quite reproducible. This is that same AAV2 sample injected multiple times on Maurice, analyzed by CE-SDS. with percent peak areas and capsid peak ratio RSDs under 7%. So the method is able to give you not only accurate measurements of the percent peak areas in your samples, but also capsid ratio, which is an important CQA. So moving on to some preclinical AAVs briefly. So these are obviously intended to be more pure, and these are data that I had presented at CE Pharm. So these are 4 preclinical AAV samples, and we can detect a number of peaks, both low molecular weight, in the middle of the peaks of interest for the VPs that I've been describing, and then a series of high molecular weight species as well. This method was able to detect a 0.1% impurity for that high molecular weight 1 species, as well as a 0.8% peak, which was only present in S2, which is shown here. So between VP3 and VP2, a novel peak showed up with a fairly reasonable percent impurity. It's a novel peak that needs to be followed up and investigated for sure. But the technique was able to measure the purity as well, which, as you can see, was over 90% for all 4 of these samples. Okay, lastly, moving on to our ICIF technology on Maurice. So same instrument, different mode. Maurice is also the gold standard technique for charge heterogeneity analysis. It's a really fast method, about 5 to 15 minutes per sample, so it's significantly faster than ion exchange, and you can run up to 96 samples in a batch. It has flexible detection capabilities, including absorbance, native fluorescence, as well as an optional blue fluorescence at 458 nanometers. Similarly, the reproducibility is great, with RSDs typically under 10%. And we've highlighted an application note here again that has put a lot of our AAV work into one nice piece of content for you. I'm going to highlight just a little bit of that work now. We have essentially 2 different methods that we use when we're looking at AAVs. The first one I call a capsid protein characterization method, and the second one is the AAV particle characterization method. They have different approaches and goals. The capsid protein method is meant to denature the AAVs, break apart that capsid, and look at the individual VP proteins, whereas the particle characterization method is meant to be more mild, maintain solubility of the AAV, but without breaking it apart. Both methods are pretty fast, under 12 minutes, and use very little amounts of AAV, between 2 and 5 microliters at 1 × 10⁻³ typically. We leverage both the absorbance and native fluorescence detection capabilities of the instrument. So shown here is a snapshot of what these 2 methods can produce. On the left is the capsid protein method for AAV6. And you can see 3 distinct peaks, 1 for each of the viral proteins. On the right is the same AAV6 sample run with our intact method. And you can see there's a fairly dramatic change in both the peak profile and the isoelectric point of the intact particle. So we've used this denatured method on those same preclinical samples I showed earlier, and again, data that I had presented at CE Pharm here. So we were able to report up to 15 unique peaks for these samples across different amounts of empty and full, as reported here below from the collaborator. We can easily quantify these peaks on a per peak basis. But realizing, of course, that 15 peaks is a lot of peaks to monitor, you could take the approach that a lot of our biopharma customers take and mainly look at acidic, main, and basic groupings of these peaks for a little bit of a broader view of the overall changes you might see. So the intact method, I'm really excited to show you this as well. This is relatively new data from us. So the intact method, again, we're trying to keep the AAV intact here. We purchased AAV9 commercial empty and full samples. And this is a picture of the absorbance shown in blue and the native fluorescence shown in black for that sample. So the absorbance at 280 nanometers is expected to capture both DNA and protein signals, where our native fluorescence will only detect the protein. Okay, that's important for this observation I'm going to describe. In this particular example with the 91% full, you have essentially a perfect overlap between the absorbance and native fluorescence channels. However, when we look at an AAV9 empty sample, was essentially empty, 7% full, you can see the absorbance, which is shaded in orange, is significantly lower than the full sample, whereas the black, the native fluorescence tracing is roughly the same. We can quantify these peaks. So this is the native fluorescence quantification on a per peak basis. And then the very last, the last entry is the total native fluorescence for both. And you can see it's essentially the same between the two. However, when we quantify the absorbance, it's apparent that there's an 8-fold difference between these two samples, which is not too far off of what the manufacturer provided. So we're really excited about what this may hold for the future in empty-full analysis. We've also proven that these methods are useful for stability indication. To induce rapid changes for the protein capsid method, we stress AAV6s to 95 degrees for 5 minutes compared to a reference sample. And we're able to see a number of changes in growing of acidic species, and then we also saw decreases in both the main and the basic species. And these are indicative of stress changes, so the denatured method works well for this. And similarly, the intact method is suitable for looking at particle stability. AAV particles have a melting temperature anywhere between 50 and roughly 80 degrees centigrade, depending on the time of the exposure. And so what we did was we took these AAV6 particles and incubated them for 37 degrees over the weekend, which is shown in orange, 50 degrees C for 10 minutes, or 60 degrees C for 10 minutes, and compared them to reference. And 3 out of those 4 traces overlay perfectly. The only one that's outlying is the 60 C for 10 minutes. The AAV is completely fallen apart, and we don't see it showing up anymore. So both methods have the ability to monitor AAV stability. So I want to come back to this pathway and again, where we've populated the instruments and just add in a little bit of the data from today. So we've been able to show that in early stage, the Simple Western platform will be useful for in-process samples because you can look with an immunoassay at fairly dirty samples. And as you move downstream, the Maurice platform with its CESDS and denatured method can look very carefully at purified proteins. And then lastly, we're quite excited about, with intact analysis, able to start understanding empty and full. And with that, I'd like to thank you again for your attention, and I'm going to pass it back to Colin.
Thanks so much, Chris, for a great talk and a really nice overview of our Simple Western and Maurice platforms. I'd like to thank Kiren again for joining us today, as well as the audience for coming to listen to our talk. And with that, I'll conclude our session.